Hello all, we are back with the next part of DNA structure and function, and in this video I will try to explain how DNA is present in different forms itself and how it associates with different types of proteins in different life forms to form what we call as chromatin or chromosome. When we talk of prokaryotic cells, the DNA is usually present with a very little amount of protein. However, in eukaryotic cells, the DNA is present in the form of chromatin with a considerably high amount of proteins that associate with this DNA to form very unique structures. And another difference between prokaryotes and eukaryotes is that the DNA is present as a circular molecule in prokaryotes, whereas in eukaryotes it is present in the form of linear chromosomes. This difference then leads to many other differences that relate to the structure and function and regulation of genetic material within the chromosomes. The linear structure is maintained with the help of special sequences at the end, which are called telomeres, and these sequences they are held together with certain constrictions. The primary constriction being called centromere, which helps the chromosomes align onto the spindle fibers. However, we should unfold this information further as we move through the slides. So DNA, when we talk of its primary structure, the linear sequence is a polynucleotide chain. Two polynucleotide chains coil around each other to form the double helix. This double helix then interacts with the histones to form the tertiary structure, secondary and tertiary structures. The uh, most fundamental structure which forms the basis of chromatin structure is called nucleosome. The nucleosomes are the uh, units that are formed by the association of DNA double helix with histone proteins. And then these nucleosomes, they coil around each other to form further condensed chromosomes, and we shall see this condensation now. What a nucleosome is. By definition, these are the globular subunits and that are repeated in the chromatin. And if we see this single nucleosome, it actually consists of this linker region between two adjacent nucleosomes and the two coils of DNA that are wound around the histone octamer. This histone octamer means that it consists of 8 molecules of histone proteins which form the core that provide a scaffold for the wounding of this DNA double helix. But it should not be confused by using the word scaffold only. That these histones not only provide the structural support and act as a scaffold, rather these are very important regulatory structures as well. Histone modifications, they play a huge part in epigenetics, which means that even if the sequence of DNA moved around the histone proteins is unchanged, the expression of the gene can be changed depending on whether it is tightly bound with the histones or it is loosely bound with the histones. These nucleosomes, they continuously join each other to form this structure which is called as beads on string apparatus. The histone octamer consists of 8 histone proteins, two of each of H2A, H2B, H3, and H4. Now, if we try to visualize the compaction of DNA in a eukaryotic chromosome, so this is a very unique structure and it has been extensively condensed. And if we try to unfold this conversation, let's start from the DNA. The DNA, the double helix of DNA, it moves around the uh, histone octamer to form nucleosomes. Nucleosomes, 3 such strands of nucleosomes, they align on each other. This basic nucleosome based on string structure has a diameter of 10 nanometer. But when three of these strings they align over each other, this forms a 30 nanometer fiber. The 30 nanometer fiber coils extensively to form loop. One loop consists of approximately 75,000 base pairs of DNA. These six such loops they form one circular structure which is called a rosette. One rosette consists of six loops, and then 30 such rosettes they align with each other to form one coil, and these coils, approximately 10 coils, such coils are present in the two chromators each. So it can be considered analogous to how a telephone cable can coil around each other, twisted around each other to form a condensed structure. There's a term which is called coil, and there's another term which is called supercoil. Supercoil means when the tension in the string increases because of rotation in one side, this results in the production of supercoil. Now, if the two DNA strands they are held together by a hydrogen bond, this means that if this hydrogen bond breaks down, the DNA strands they can separate. This process, where two DNA strands they separate from each other because of hydrogen bond breakage, is called DNA melting, and each of these single strands is now capable of binding to a complementary strand, whether with themselves again or to another complementary sequence, and binding of any DNA strand with the complementary strand is called annealing. So let's see. For example, this is a double helical DNA, and you have treated it with heat or with a chemical which results in separation of these two strands from one another. This separation is called melting or denaturation. But depending on the size of DNA, not all part of DNA may be melted or denatured. So it may retain the portions where DNA is still in double helical form, and it may have portions where DNA is in single-stranded form because of hydrogen bond breakage between them. So such a DNA molecule is called partially denatured DNA. Now, what can happen that this molecule, if it is further treated with the same chemical or heat, it may result in complete separation of the two strands, and if not followed up properly and not uh, getting into the proper downstream process, these strands they can anneal with each other to form again the partially denatured or the completely renatured DNA molecule, which will be called renatured because the same DNA strands have annealed within themselves. So the melting point of DNA is determined by heating the DNA and then seeing where half of the DNA, 50% of the DNA is denatured, which means that this is a DNA molecule which has half um, uh, DNA which is still in double-stranded form and half DNA which has been denatured or converted into single-stranded form. That is the bond between them, hydrogen bond between them has broken down. So for different DNA molecules, the melting temperatures can be different. That is the point at which 50% of them is denatured can be different. For example, for this DNA molecule, it may be around 82.5 or 83. And for this DNA molecule, it is around 85. Why there is a difference between the melting temperatures of different DNA molecules? Because certain their composition can differ. For example, a DNA with high GC content will require more heat to be broken, as GC pair has three hydrogen bonds, so more heat is required to break three bonds compared to an AT pair where only two bonds are present. If the size of DNA is large, of course, more heat has to be given. pH is an important factor because DNA is a polar molecule hydrogen bonds they produce, they are dependent on the electrostatic interaction between hydrogen protons and electronegative atoms. So pH is a very important factor that determines the melting temperature, and the ionic strength of the medium in which the denaturation is being performed affects the melting temperature of DNA molecule as well. So what we have done till now, if we try to summarize the DNA function and structure, what we can say that DNA consists of four bases: adenine, guanine, cytosine, and thymine, that are held in a linear array. This is the primary sequence of the DNA, which is made by the phosphodiester bonds between two nucleotides, bases of these nucleotides, between 3' hydroxyl and 5' phosphate group. These each when a strand forms two such strands, they are held together by pairing of nitrogenous bases, and there are two hydrogen bonds between adenine and thymine, and three hydrogen bonds between guanine and cytosine on the complementary strand. And these strands then form the double helix around the central axis, which is also called the diaxis of central symmetry. When we talk about humans, because we are humans and we are interested in the information related to humans, so there are approximately 3 into 10 to the power 9 base pairs of DNA in humans, which is organized into a haploid content of 23 chromosomes, which means that this content, this DNA corresponds to the haploid content. And DNA provides a template for its own replication and also for the transcription. When it is a template for its own application, it maintains itself. It maintains the genotype of the organism, and when it acts as a template for transcription, this means that it results in the production of roughly, in the expression of roughly 30,000 genes, which then I can either result in the formation of active nucleic acids like transfer RNA or ribosomal RNA, or messenger RNA, which then translates into various kinds of proteins. With this, we end today's lecture, and I hope you have enjoyed. Thank you very much.